tens of thousands, like you would be with mastodons or something. They can get fossilized DNA out of some of those, and it's still useful. But it's definitely, I was trying to get mushroom DNA out of mushroom samples that were like 5 to 10 years old, and I was having trouble. So that's 5 to 10 years. So yeah, I would know nothing about this, but there's a whole set of techniques that are developed around working with ancient DNA and getting DNA out of fossils. There's definitely limits on it, and it's much more difficult than working with something that's fresh, but yeah, they can do it. Most of the phylogenies that you see, though, that incorporate fossil taxa are being done with morphology. As far as I know, very few people would be doing molecular phylogenies with fossil taxa. Okay, so the Xenarthrans, if we look at that little group, this group right here, should have pointed to it. It would be things like lagomorphs and rodents and presumably primates. But like I said, I think this is kind of controversial, especially the placement of primates within mammals and what the closest relatives to primates. I think that's the thinking on that has changed quite a bit in recent times. I don't think it's anything that they're totally settled on. There's not a lot of strong support to put primates in any one particular group. And then if we looked at sort of that last huge group that they have down there with all those different orders together, these would be carnivores. So things like pangolin, as weird as that is a derived carnivore. These are pinnipeds, right? So have I convinced you that seal's a dog? These are perissodactyls, right? With the odd-toed hooves. Then these are the set artiodactyls, so things that have even-toed hooves, and then all the cetaceans, so toothed and baleen whales. Notice that a lot of people, I think that whales are related to like seals. That's not right. You realize that's not right. They're pretty distantly related. And certainly they both have gone back into water independently of one another. They're not each other's closest relatives at all. The closest relatives of cetaceans are things that are terrestrial. The closest relatives of pinnipeds are things that are terrestrial. And then there's a big group of chiropterans, right? Okay, so here we go. Which mammal is most closely related to a seal? Okay, come on. I just said this like 10 times, right? A horse, a monkey, a dog, or a whale. In fact, I just said it like 2 seconds ago. Okay, is that everybody? 13?
Oh no. Wait for it. There we go.
Okay, so pinnipeds are related to dogs, or carnivores at least. So which of these mammals is most closely related to an armadillo? A sloth, a walrus, a possum, or a gorilla? Remember, this is a southern hemisphere group, right? The northern hemisphere members are descended from southerners that came in.
Weren't you 17? Weren't you 17?
No, I'm not 17.
Oh.
I'm 12.
Oh.
So it's literally on the number.
Oh, nice. Very nice.
Okay, one more.
Yeah, mine's not working. Hold on.
I think
Okay, so sloth, right? These are xenarthrans, right, like sloths. And anteaters would be the other one. So, obviously I talked for a long time, but I answered one of the questions on here. That's the end of all the different organismal groups. So everything from bacteria that we started with a couple months ago, gone through all the protists, all the fungi, all the plants, all the animals right to the end. That's the end of the course. You take the exam, there's like 5 more lectures, and that's going to be ecology. So we'll focus on sort of aspects of the environment and how organisms adapt to their environment. And really spend a lot of time focusing on population conservation, population and community ecology, and a little bit on conservation at the end. Yeah, both lab next week. Ecology modeling lab the week after that, lab practical, that's the end. We're right to the very end. So what we need to do is kind of work our way through this exam, and I think we can do it kind of quickly. I'm hoping we even finish a minute or 2 early. And if this feels too quick for you, go ahead and get together some questions and bring them to me like Monday if you can, or early on Tuesday or something like that, yeah. I should probably be here most of the afternoon on Tuesday because I have to go to the high school in the morning, so Not that that's not kind of last minute. Either that or email them to me over the weekend and I'll get back to you on Monday or something like that. Because I'm gonna go kind of quickly. I'm assuming you kind of want to know what I think the answers to these are. Yeah. So let's go kind of fast. The first one says, historically, relationships between animal phyla were inferred such that emphasis was placed on Salome type, right? It now appears that Salome type is not a reliable character for inferring relationships. What two characters, or name two characters, that might be more reliable and justify your answer? So, anybody want to suggest one?
Cleavage?
So cleavage type, I think, would be a good answer. Do you know what he means by that?
You have the bilateral symmetry, then you have the radial symmetry.
Yeah.
Better than Salome type.
Oh, okay, so symmetry, I think, is a less, they said that in the other class too. I would take that definitely as an answer, but I think it's a less good answer than.
Larval stage?
Let's look just really briefly. This is what it's referring to, right? This is the old tree that would have been in Campbell's 5th edition. And you'll notice here you see things like acoelomate, pseudocoelomate, coelomate, Salome coming from a schistoseel, Salome coming from an intracoel. There's all kinds of stuff in here about the Salome type or lack of the Salome or the derivation of the Salome. And here that's completely gone, right? And so the idea would be to look on here and see something that maybe would come close to defining clades here, because you'll notice those different Salome types. You have, like this guy's acoelomate, this is acoelomate, this is pseudocoelomate, pseudocoelomate, this is pseudocoelomate. These have a true Salome. This has a true Salome maybe, but it really has a large hemocoel. It's probably a pseudocoel with a small true Salome. These have a true Salome. I mean, it's a mess, right? That character is mapping all over the tree, so you can't use that to define a group. So the question is now, looking at this tree, what characters might sort of define groups?
Doesn't all the bilaterally symmetric animals have true Salomes?
No, that's not right. Platyhelminth doesn't have a true Salome. It doesn't have any Salome. It doesn't even have a pseudocoel. And rotifers have a pseudocoel. Nemurians have a pseudocoel. Not sure about these. Nematodes have a pseudocoel.
But none of the radiosymmetric animals have true
They don't have a Salome at all. They're all acoelomate.
So technically, couldn't that be the ancestral condition, but they lost it for the?
Right, the platyhelminths lost it, and exactly where, yeah, right, so, but that's the problem, is that you have losses and gains and stuff like that, such that it's not a good character to try to define a group.
So that answers my own question, that it's not really good to classify.
It's not, right, it's not gonna be used very well to classify. There was a lot of emphasis put on that before, and we really don't use it at all now. So the question would be, you said body symmetry, you could almost do that because of radiata and bilateria, but remember that other phylogeny I've been showing you, right, doesn't actually put the radiata together. So that's a little bit more questionable. But you could argue, for example, this one, right? These would be the radiata. So radial symmetry is probably the ancestral condition, but you could argue that bilateral symmetry defines this group here. I don't think that's a wrong answer. I just don't know that it's the best one. But when you say cleavage type, right, so what are you referring to there?
The two different
That
cleavages?
you have a couple different types of determinate cleavage that you know. This one that sort of defines the Lophotrochozoans, this spiral determinate cleavage that you see in mollusks and annelids and platyhelminths at least. And then this idiosyncratic cleavage that appears to really well define ecdysozoans. So what could you put besides cleavage and maybe body symmetry? Is there another one?
Larval stage?
Larval stage to a certain extent, because you have this Lophotrochozoan group that has this distinctive trochophore larva that you don't find outside of that. That would be reasonable. What else?
Like blastopore development.
The fate of the blastopore
Yeah.
seems to be really big, right? This idea of having protostomes and deuterostomes, and those groups were recognized before. Of course, things like platyhelminths probably wouldn't have been considered protostomes, but this idea of fate of the blastopore, that appears to be phylogenetically conserved. So that's probably a pretty good answer. I think if it were me doing it, I'd say fate of the blastopore, and I'd probably say cleavage type. But these other answers that you're giving are reasonable too. So can you all do that? Could you all do two? And as far as, huh?
Could you say just, protostome development or deuterostome development, or is that
Yeah.
broad?
Oh, I'd rather you say fate of the blastopore. And then to justify your answer, say, it appears that all the animals that have the blastopore become the mouth,
Okay.
form this clade that we know as protostomes, and, yeah.
But all of the protostomes that have the determinate cleavage, you can't separate those early states because they won't
No.
survive, right?
Because here's the problem, here's where you're gonna start to see weird stuff. You realize ctenophores have determinate cleavage. Sorry.
Cnidarians and ctenophores?
Not cnidarians, ctenophores, right. Aceals, the little group of platyhelminths is potentially the sister group to the rest of the
But both of them have
bilateria.
radial symmetry though.
These have, yeah, but remember that these guys have bilaterally symmetrical elements to their bodies, so sometimes people say that they're biradial symmetry. So they're not even really completely radially symmetrical. You could debate that a little bit. You could also debate whether or not some of the
That's what you're saying.
Nigerians are maybe triploblastic. It's problematic, it's a problematic thing, and I'm not sure if this really holds, this group between the two, because it's not shown in that other tree.
But they only
But
have
as
two
far as-
layers though, right?
Huh?
Both of those only have two layers, they're diploblastic, right?
Right, but remember that there's some cells in the mesoglia of jellyfish sometimes, so some people might argue that that's actually a third tissue layer qualified as another dermal tissue. Nothing's easy, right? You realize, nothing's easy. And this business of determinate cleavage, notice that these guys have indeterminate. Pretty sure these guys all have radial cleavage, right? But you see this derived determinate cleavage, this spiral cleavage in platyhelminths, mollusks, and annelids, right? And some of the other Lophotrochozoa and phyla. And then this idiosyncratic cleavage that you see. But you don't see any, as far as I know, any kind of determinate cleavage in deuterostomes. But you do see it in some of the groups that are even outside of protostomes. It's not a uniquely protostome thing. And there are plenty of protostomes that have indeterminate cleavage. Presumably indeterminate cleavage is the ancestral condition. And these different types of determinate cleavage you see are all derived. But there's probably 12 different, even though you're just learning 2, like spiral and idiosyncratic. And if we were doing this a couple of years ago, you would have been learning 1. We would have just called it spiral. And we would have called everything that all the arthropods and nematodes and everybody does is spiral too. Now at least we're learning 2, there's probably really about 12 or so different independent acquisitions of different types of determinate cleavage, mostly within protostomes, but there's some other lineages like pinafores and stuff that have it too. So it's complex. I think I'd pick cleavage types and theta blastopore. It's up to you. Those would be good answers, I think. Okay, how about let's do lickety-split this starfish here. So the phylum is, and notice this picture's right out of the slides, right? So phylum is what? Echinodermata, the class is Asteroidea. And on the diagram, let's maybe start with the arrow that's pointing straight up, and then we'll work our way around clockwise. So pointing straight up, that would be the what? Y'all just dissected one of these, right? Huh? Stomach, it's the pyloric stomach, but if you put stomach, that's good. Okay, the next one over, what do you call that little thing? The madreporoid is the pore, the seed plate, either one of those would be fine, I'd take. How about the next 2 little lines down, what are those? Gonads, right? How about these next 2 little lines down, they're pointing to what? 2 feet would be good. If somebody put movable spines, I'd take that too, but 2 feet I think is the better answer there. How about next line over, the single line? Radial canal, right, it's pointing to that canal that's moving down underneath the ambulacral place. How about the next line up? Ring canal, right? And then these next 2 little lines that are pointing down would be what? The digestive cechi, right? And over here in this little picture off to the side, the thing pointing to the little water balloon, what's the little water balloon called? Ampulla, right? And then it says, what's the name of the surface of the starfish that's visible in this picture? So which surface is that? Remember, we don't really say anterior, posterior, dorsal, ventral. It's the aboral side, right? It's the sort of side that's facing away from the mouth. Yeah? Notice that's just a bunch of labels. That would be easy to fill out, maybe.
In these lectures, you pointed out like this one. You pointed out about like 7 or like 10 different slides that we would have to remember. That like,
Yeah,
do we have
good.
to remember
Any
the
slide.
7? Any
All
slide?
slides.
There's a
That
lot
narrows
of
it down. There's
labeling.
tons of pictures that are in the book that aren't the slides.
Yeah.
You know the slides, that would be pretty good. Seldom would I use a picture that's not.
With the labeling aspect.
With the labeling, definitely. I wouldn't ask you to label a picture unless I've shown it to you in the slides.
There's like 8 or 10 of them that you pointed out.
Yeah, that's good. Yeah, labeling, I wouldn't ask you to label a picture unless it was in the slides.
Okay.
How's that? I might show you a picture and ask you a question about it that wasn't in the slides, although that's very rare. But I would never ask you to label one unless I showed it to you in the slides. Okay, so here for the clam, I already showed this one, right? The phylum is Mollusca and the class is Bivalvia, right? As far as getting the anterior, posterior, dorsal, ventral on those 4 lines, first class I gave this to as an exam question was a train wreck, right? Because they couldn't figure out. The problem comes from the fact that it doesn't have a head, right? But that doesn't matter. You just figure out where the mouth is. Hopefully you all realize the mouth is on the left-hand side here. Here's the anus over here. If you weren't 100% sure, you might remember that the siphons are on the posterior end of the animal. We did dissect one of these in the lab, right? So this is anterior on the left, posterior on the right. And then as far as dorsal ventral, you might not be super sure. You might have remembered that the hinge side is the dorsal side of the animal. But otherwise, the other little giveaway might be that the heart is there. And remember, the circulatory system in invertebrate animals is dorsal, right? Your circulatory system is ventral, but theirs is typically dorsal, and their nervous system is typically ventral. It doesn't really have an organized nervous system, but the circulatory system being dorsal might give it away. Yeah? So dorsal on the top, ventral on the bottom. So do y'all get that? Anterior, posterior, dorsal, ventral on those 4 lines. Then it says, using only the characters labeled in the diagram, list 3 synapomorphies for the phylum. So what labels on there give away that it's a mollusk? Go ahead, pick one. Foot, I would take. Foot, I would take. The type of foot, the type of muscular foot that you see in a mollusk, remember a mollusk is supposed to have a 3-part body plan, a head, foot, a visceral mass, and a mantle that secretes the shell, right? So foot, I would take, I think that's a reasonable answer. How about anything else on there?
Mantle.
Mantle would be very good. What did we talk about outside of mollusk that has a mantle? Nothing. And they all have, from a squid to a snail to a clam, they all have a mantle that secretes a shell, right? So that would be a good one. How about what else could you put on there? That's, Ctenidium would be good too, right? As long as you're calling it a ctenidium and not a gill, that particular type of a gill structure, that's unique to mollusks, right? I didn't call anything outside of mollusks a ctenidium, only that. And again, you see it in everything from a conch to a clam to a squid, right? And what's the other one you want to put? Well, I don't know about Salome, because Salome's not uniquely molluscan, right? So for example, annelids have a Salome too. Trying to find something that defines mollusk, that really makes you know it's a mollusk. Which one?
Nothing.
You're picking names off this picture, right? It says using only the characters that are labeled in the diagram.
Anti-detertion? Isn't, no?
Are you picking things on the diagram?
Detertion, isn't that something that they do because
Torsion,
of
that's
torsion?
for gastropods, not for mollusks, so. And it's not on the picture. You're picking these words up here. Start making up other stuff. Pick these words.
Oh, you're picking words.
So mantle we've got, foot we've got, ctenidium we've got. Those are 3 good ones. Shell, really. What else did I show you that has a shell? None. Maybe a brachipod, but we didn't really study brachiopods, and that's a different type of a shell than what's here. You realize crabs don't have shells, right? They have an exoskeleton. They have an exoskeleton, right? Not a shell. So this type of a shell that we see in mollusks, that wasn't in any other group, right? I think those are probably the best one. Things like adductor muscle and stuff like that, and siphons, I'd probably steer away from those. Those, because you're going to find those in bivalves, but that's not a phylum-wide character. How about, like, does it have a radula? No. Remember, bivalves don't because they're filter feeders. They've lost a radula, presumably. Okay, so how about number 4 here? We've got a little phylogeny of arthropods. So you see there's those dark lines, and it asks you after each one of those dark lines to write how many pairs of antennae that group has, right? So myriapods are what? Is there a picture in your head? I'm afraid there's not a picture in your head. Centipedes and millipedes. How many pairs of antennae do they have?
1, 2,
Pairs.
1.
1. Chelicerates. Who's that? Oh my goodness. Horseshoe crabs, spiders, scorpions, ticks, mites. How many pairs of antennae do they have? 0. So you've written 1, 0. How about brachiopods? Who's that? Is there a picture in your head? Sea monkeys, Daphnia, right? How many pairs of antennae do they have? They're crustaceans. Crustaceans, as historically defined, not including insects, all have 2, right? And then malacostricans, who's that? Dissected one. Crayfish, crabs, lobster, shrimp, isopods, right? They all have 2. And insects all have 1. So then it says, talk about the evolution of that character. So here I want you to talk about convergent, something you could use as a synapomorphy to define a particular group. Hopefully you see you have 1, 0, 2, 2, 1, right? It's kind of a mess. Remember, historically, we would have wanted to put myriapods and insects together because they have a single pair of antennae and unbranched appendages and spiracles and trachea and all that kind of stuff. That doesn't really work anymore. Notice that myriapods and insects are together in this tree. The reality is that there's probably convergence here. So you don't really know what the ancestral condition is, for example, for myriapods and chelicerates, what the ancestral condition would have been there. It might have been to have no antennae and then myriapods got 1, or it might be that the ancestral condition is to have 1 and chelicerates lost that 1 pair.
Could even be 2.
There could even be 2. Maybe if you go back to the very base of the tree, it was 2. and then myriapods lost one of the two pairs, and chelicerates lost both of them,
Yeah.
and then insects lost one. So maybe the ancestral condition for all arthropods is to have two. Maybe the ancestral condition is to have just one, and you gained one in the arthropods, in the crustaceans, and you lost one in the chelicerates,
We
right?
might have none.
Or maybe it could have been none. And right, and so chelicerates are showing the ancestral condition, and myriapods gained one, insects gained one, and the crustaceans all gained two somehow. That's what I want you to say over here. It's a mess. There's no easy way to construct what the ancestral state
As
would
long as
be.
you have one of those in each plant.
Yeah,
you're good.
right. You don't have, it's not like you have all the guys with one coming out together in the tree, and all the guys that have two coming out together in the tree, and all the guys with zero coming out together in the tree or something. You got it scattered all over here. So clearly, there's got to be some convergence and retention of ancestral conditions with respect to numbers of pairs of antennae. Okay. 5, looked at several animal phyla that have both free-living and obligatory parasitic members. Choose 2 different phyla that meet this criterion. And then name and briefly discuss the biology of one specific free-living and one specific parasitic member for each phylum so that you can convince me your answer makes sense. So name a phylum that has both free-living members and parasites.
Nematode.
Okay, nematodes would be a great one. So name a free-living nematode. You look at
Heartworms.
Huh?
Heartworm?
No. Nematode.
Is it not
An animal.
Oh.
Well, that's a good example of a parasite, but that's not a nematode. Okay, so that doesn't work. Parasitic nematode? I said a free-living nematode. You gave me a parasitic platyhelminth. Give me a free-living nematode. I only have one live nematode for you in the lab.
What was the one in the movie?
The one in the movies, that's a platyhelminth. That's a flatworm.
Oh.
Yeah, so hang off on that. Let's have that be our other phylum. So we got nematode. You guys are confusing nematodes and platyhelminths.
Yeah.
How about nematodes? How about C. elegans?
Oh,
Remember the
that
little
dude.
ones that were in the little plate? They were crawling along like little snakes? You were all looking at them? No?
Small
Maybe.
little worms?
The little tiny ones that you couldn't see without looking under
Yeah,
the
the
dissecting
tiny
stuff. They were on that plate. Those are free living, right? They're free living. They're eating bacteria that are on the plate. So C. elegans would be a good example of a free living nematode. How about a parasitic nematode now?
Heartworm.
Don't say the one in the movie,
No.
huh? Trichinella would be a good one, the one that causes trichinosis, the one that causes elephantiasis, right? How about those beautiful heartworms that were in the dog? Dog heartworms are parasitic nematodes, right? There's a bunch of them. Okay, so nematoda would work. Now let's try platyhelminthes. Okay, so if you said platyhelminthes as your phylum, name a parasitic member, now you can say schistosome, right? Or blood fluke, or liver fluke, right? Or something like that. How about free-living platyhelminth? Huh? Oh, no, that would be a parasite still. So you could have said tapeworm, too, would be another example of a parasitic flatworm.
Was it brown worm? What is it?
Well, that
What is
little
it
brown
called?
guy with the little...
Earthworm? What is it?
No, no. Dugassia would be
Dugassia.
a great free-living one. The little guy that was eating the egg yolk, right, with the little eyes on his head. You forgot him already.
Yeah, I
The
forgot.
cute little auricles, those little ears.
Oh, Naris. How about that?
No, that's a different one. You keep naming annelids. Now, there might be parasitic annelids. Okay, so let's imagine you said annelids. So you'd say nematode and platyhelminth, right? If you said nematode, you name a free-living and a parasitic one. If you say platyhelminth, you name a free-living and a parasitic one. But let's imagine you had said annelid. So for annelid, free-living, you say Nereus, right? That's your free-living one. Or you say earthworm. What could you list as a parasitic annelid? Something that latches onto you and sucks your
Oh,
blood.
leech.
Maybe a leech or something like that, right? There are potentially other phyla you could do this for too. I think those are the easiest ones. How about if you did Arthropoda? Could you do that? Yeah, like a tick and a spider would be free living. It would be a predator, but it's not parasitic. But there, I think I would say Arthropoda, rather than narrowing it down to what you said is basically a class, Arachnia, which is good. You can even get inside of a class and have it still a parasitic and a free living member. But if you do it, just make sure you name a phyla, because it says name 2 different phyla.
Now, when you say discuss the biology, do...
That, yeah, very briefly. So leeches suck blood or something. I mean, you could give me more if you want to, but don't get too crazy. Yeah.
So what does heartworms do? I mean, I know they kill the dog, but...
Yeah, I'm not going to have to go into more detail because I'm not going to be able to. They live in the heart and they kill the dog. They're parasitic on the dog. That's good enough for me. That's their biology. I'm more interested, can you actually, first off, make sure you name a phylum, right? The only
Yeah,
phyla
that's
that we
what
have
I-
here are Porifera, Cnidaria, Ctenophora, Platyhelminthes, Mollusca, Annelida, Nematoda, Arthropoda, Echinodermata, and Chordata. Those are the phyla, right? So just make sure you name, if it says something like choose 2 phyla, maybe on your cheat sheet, can you just write down the 10 phyla that we covered? So when it says choose two different phyla, you don't give me something that's not a phylum, because that's gonna make me cringe, right? You can't give me something like Trematoda as your answer, because that's not a phylum, right? Give me a phylum. At the very least, give me a
I was
phylum.
just saying, the heartworm that's in Nematode, the heartworm would be an organism that's in the phylum Nematode,
Right,
and it's parasitic.
right, exactly. And C. elegans would be a good example of one
Discuss the
that's.
biology of the phylum, is that what we discussed biology?
No, of the
Biology
animal, of the specific animal, yeah. Name and discuss the biology of one specific member, yeah. Okay, so closed circulatory system means what?
Same exit for
No,
an eating?
No,
They
that's
don't
closed
have-
digestive tract.
Oh,
The
Oh, like
blood-
us.
The blood is in
That all the blood's contained inside of blood vessels. There's no blood loose inside of the hemocoel, right? And obviously, it says then which type do you have? You have
close.
closed. Two invertebrate lineages that have closed. Most invertebrates have an open one, right?
Mm-hmm.
Okay, well, what did you keep saying as a phylum?
Who,
A
me?
second ago, yeah.
As a phylum?
What animals did you keep naming that kept shooting you down? Annelids.
Oh, okay.
You kept saying Urius and earthworm and stuff like that. Annelids, right? And then cephalopods. Squid, octopus and stuff. That was sort of closer to this.
Those are the two invertebrate lineages?
Yeah.
What was that?
Annelids and cephalopods.
Annelids and cephalopods.
Okay, then we come to 7, which I'm going to do lickety-split here. Let's do 8 first. So 8 says that the evolution of the amniotic egg is analogous to the evolution of the seed in terrestrial plants. Why? What does a seed do? The seed pollen habit freed terrestrial plants from what? A need for water in order to reproduce. The amniotic egg does something very similar for vertebrate animals. What group would be analogous to non-seed plants? What group within the vertebrates would be something like non-seed plants? In other words, something that's terrestrial but still tied to water to reproduce. Amphibians, right? And what group would be analogous to the seed plants?
Mammalian?
Mammals and reptiles and birds, right? The amniotes, basically. Okay, did you all get an answer for 8? Right? Okay, so now we come to 7. I love this question. I'm going to go through it really quickly with you here. Can't ask a question like this on an exam because I'm afraid none of you get any of it right. And the reason is because what it has is a whole bunch of names over here, and then it has a whole bunch of different characters or structures that refer to some aspect of that biology. So the idea is to match one of these names over here, and each one takes only one. So notice that even though a couple of these could go in a couple different places over here, you kind of by process of elimination could figure out each one only goes in one spot. And I'm going to do it with you really quickly right now. The first time I used this on exam, it was just horrific. And what I realized is that they didn't even know what the names were. So for example, when I'd say a name like Cnidaria or Trematoda, they couldn't even picture an animal, long let actually know what the structure looked like and be able to tie it to a particular group. Yeah? On your exam, on every real exam I've given besides these practice exams, I'm going to give you this, 20 names. All you got to do is give me an animal. Picture in your head. So I'm going to say Scyphozoa, you're going to say jellyfish. I'm going to say Trematoda, you're going to say, not saying anything. You're going to say schistosome or blood fluke or something like that. I'm going to say Holothuriidea, you're going to say sea cucumber, right? I'm going to say protostome, you're going to say, it could be a big group. You're going to say what?
Platyhelminth?
Platyhelminthes works as a protostome. You could have, but I don't want you to say platyhelminths. I want you to say an animal, a specific, exact animal. So you could say Dugassia, that would work for me, right? Or you could say schistosome, or you could say tapeworm. Don't say worm, that doesn't fly. Don't say fish, that doesn't tell me anything, right? It's got to be the more specific you can make it, the better. I say hemipteran, you say? No, that's orthopteran. At least you know it's an insect order, that's good. No, that's a coleopteran, huh? Hemipteran. How about cicada, aphid? Lordy. There are about 200 plus names I could pick from. Every single name, everything from ecdysozoa to protostome to bilaterian, ctenophore, every name you could imagine. Gnathostome, vertebrate, chondrichthyon. Think of every name I've given you related to an animal. I'm going to pick 20 of them. All you've got to do is just name an animal. That's it. And you get 20 points. But you have to name an animal that's actually in that group. And it has to be a very specific animal. You can use The genus name, if you want to, you could put, if I put Nematode, you could put C. elegans if you want. Right? But you have to name a specific animal. You're all going to practice for this, right? You got to somehow get these names. So let's try this. Arachnida would be what? Spider. How about mollusca? Or you could have said tick, scorpion, right? How about mollusca? Name an animal. Clam, snail, squid. Loligo, you could say, right? What's that? Squid. That's the squid you dissected, right? But you don't have to. You can just say squid if you want to. Trematoda. What are you going to say?
Schistosome.
Schistosome. Or blood fluke are probably the only things you could say. How about Cnidaria? You're going to say what? Jellyfish. Or you could say sea anemone, coral, hydra, bisalia. Right? What's bisalia? Portuguese man of war. Mammalia. I did this the first time people left that blank. Yeah, dog, cat, right? Panda. I mean, come on, there's a zillion mammals. Some people put their own name. That's cool, right? But, yeah, come on, mammal? I was like, when I gave it back, I was like, come on, guys, how do you leave a mammal blank for crying out loud? Okay, Meristomata, the only thing you can put is what? Horseshoe crab. Or Limulus is the genus name for it. Hexapoda, who's that? That's insects. Fly, bee, whatever, right? Oligochaeta, there were 4 we looked at in lab.
Dora.
Dara. Lumbricus, Elosoma, Tubifex, or you just say earthworm would work. Echinodermata, who's that? Starfish, sea cucumber, sea urchin. Myriapoda, who's that? Centipede or millipede. Cestoda, who's that? Tapeworm. Maxillopoda, who's that? Those are Malacostrica. Maxillopoda are copepods, like cyclops that you looked at live and in person up there, and barnacles, right? How about Polychaeta, who's that? With mini chaetae. Nereus,
No.
right? Or you could say tube worm, you could say pile worm, clam worm, don't just say worm, yeah? How about Porifera, who's that? Sponge. Myxini, who's that? Hagfish, right? Bivalvia, who's that? Clam, oyster, mussel, right? Malacostrica, now you can say crayfish, shrimp, crab, lobster. Amniote, what's that? A bird. Well, yeah, don't say bird, say hummingbird, right? Make it specific, say chicken, right? Say boa constrictor, right? Say something specific. Hirudinea, who's that? Leech, right? How about Odonata, what's that? Means toothed mandible. Those are damselflies and dragonflies. Let's go really fast through the characters here. Radula, what's that? Is there a picture in your head of what that even is?
Tongue.
It's a little tongue with teeth, and you associate it with who? Mollusks, right? So it would go on the mollusk line. How about chelicerae? Who do you associate that with? That's the first pair of appendages in chelicerates, but we have arachnids up here, and we actually have Meristomata too. It would be in both of those, right? In a horseshoe crab or in a spider or scorpion. So chelicerae, we don't know whether it goes on arachnida or Meristomata, so put a little check mark over here. We're going to have to hold off on chelicerae. How about ampulla? What are those? The little water balloons that are part of the water vascular system of an echinoderm, so that would go where? No, echinoderm. How about echinodermata? Single pair of antennae, who's that? Could be Myriapoda. Who else could it be? Who else has a single pair of antennae? Could be Odonata, maybe even. Who else could it be? What's the larger group that Odonata belongs to? How about Hexapoda? Those are insects, right? So we don't really know where that goes. Put a little check next to single pair of antennae. How about choanocytes? Those are the color cells that you find in a what?
Sponge.
Sponge. So those go under Porifera. How about nematocysts? Those are the stinging cells that you associate with a
Hydra.
hydra, which is in which phylum? Hydra's not.
Hydrozoa.
No, that's the class.
Oh.
Cnidaria, right?
Cnidaria.
So nematocysts go with Cnidaria. How about Redia? That's a hard one. It's one of the larval stages, if you look in the lab manual, of a liver fluke. Where are flukes on here? What class are flukes in? Trematoda. So Redia goes with Trematoda. How about Scolex? You all drew one of those, supposedly. Those are the suckers and the hooks that are on the end of a tapeworm. It goes Cestoda, right? How about Parapodia? Those are what? Little feet with the CT, right, on Nereus. Nereus is a what? Polychaete, right? So Parapodia go with Polychaeta. How about who has an incurrent siphon? It filters water. How about bivalvia is the only one I think on there, right? How about a clitellum? What's that? Do you know?
It's in the earthworm.
It's a little Band-Aid on an earthworm, right? So what's an earthworm? Where does it go? A Ligoceta. So clitellum goes with a Ligoceta. How about anterior sucker? Who do you think that's on? Who's going to have a sucker? A leech. So where is that? Iridinea. How about slime glands? Where does that go after that movie?